Hello everyone, welcome back to one more video session. In this session, I'll be speaking about very important concept, could be asked for 5 marks. That is Watson and Crick's. That is James Watson and Francis Crick's famous double helix model of DNA. They proposed this in 1953 based on the X-ray diffraction of DNA. We'll write here. You get this question for 1 mark. On what basis Watson and Crick proposed a double helix model of DNA based on X-ray diffraction of DNA. We'll write here X-ray diffraction of DNA by two scientists. Prior to this proposal, the two scientists, let me write the name of the scientist. One fellow is Astbury and other famous lady. She is we know about her, that is Rosalind Franklin. These two scientists, they were working on DNA, that is they took the photograph of DNA based on X-ray diffraction. First, it was conducted by the Astbury, male scientist, whereas after that another female scientist, that is Rosalind Franklin. First Astbury said that the distance between the base pairs, that is that, let us say adenine, thymine, and here guanine, cytosine. In a DNA, you see the base pairs throughout the DNA. The distance between these two base pairs is 3.4 Armstrong units. That's it. 3.4 Armstrong units said by the Astbury. And after that, he also said that in the DNA structure, you see these nitrogenous bases are actually stacked like coins. Remember this? I think better I'll draw the diagram. See here, these are all the nitrogenous bases. If you look into the DNA, let us assume that this is the backbone of the DNA, that is pentose sugar and phosphate group, and these are all the nitrogenous bases. If you look here, it looks like many coins are stacked. This is actually proposed or said by the Astbury. Just remember, he said the distances between the nitrogenous base pairs will be 3.4 Armstrong. The interval between these two nitrogenous bases is 3.4 Armstrong. At the same time, he said the nitrogenous bases are like stacked coins in a particular DNA based on his X-ray diffraction. It was proposed before 1950. And after that, the Rosalind Franklin, a famous lady scientist, she actually also conducted the same experiment. That is, she took the photograph of the DNA based on the X-ray diffraction and said that the DNA has some sort of helix, in the sense that DNA is double helix, right? She said the DNA has some sort of helix like structure. Helix, what is helix? Like this. But she did not propose the double helix model of DNA. She had all the chances to propose it and she could get the Nobel Prize. Unfortunately, let me be frank, even one more scientist, actually the colleague, co-worker or co-scientist of Rosalind Franklin, called as the Wilkins, actually stole her X-ray photography. That is, the truth actually happened. Stole the X-ray photography of the DNA, and then this model, it was actually given to these two people, that is Watson and Crick, and they stole the information of Rosalind Franklin's work and they proposed this DNA helix, and they got the Nobel Prize. That is, Watson, Crick and the Wilkins, this cunning fellow, got the Nobel Prize in 1963 after 10 years. But this lady, the Rosalind Franklin, she was actually very much interested, fascinated towards finding what is there exactly inside the DNA. She was working on DNA and she had all the chances to propose the double helix model of DNA because she knew it. Yes, this is the structure of the DNA. She knew everything about it. Okay. But unfortunately, as she was working, obviously she has to use the X-rays and everything. She got cancer. She died of cancer in 1958. And after 5 years, these three people, including this Wilkins, got the Nobel Prize for their work, for the proposal of double helix model of DNA. This is just one information. Don't write it in the examination, just information. Okay. So always remember, Rosalind Franklin is a role model for majority, all the lady scientists. And in those days, the lady scientists were not given that much of importance compared to the males. It's like it was male dominated age. Okay, fine, chalo. That's it. That's enough. Now we just focus on the Watson and Crick's double helix model of DNA. Here I would like to give one more information, that what kind of DNA model they proposed. This is very important. They proposed BDNA. What is this bDNA? It's just a name given to the DNA proposed by the Watson and Crick. So please remember, Watson and Crick, they actually proposed B form of DNA. This is the B form of DNA. In DNA there are 4 different types. This is for your need. There are 4 different types. I'll tell you why all this classification is made. This is first we write A DNA. And the other one is the bDNA. This is the famous and commonly seen in all living organisms. In my body also I have this bDNA, which is like 95% of the DNA is bDNA. Remaining, I'll tell you, it could be zDNA. The third type is called as the cDNA. Don't expect me to write the DNA. It is zDNA. So A, B, C, zDNA. Okay, I'll tell you what is this aDNA. It is normally not seen in all living organisms. Very rarely you see them. Sometimes, like when you pull the DNA, sometimes you see aDNA. What is this A, B, C, D or zDNA? There is no dDNA. What is this? This is nothing but when you look into the structure of the DNA. Okay. So this is the structure of the DNA. These are all the nitrogenous bases. If you count, this is actually the helix. That is the initiation of the helix, the starting point of the turn, and this is the closure of the turn. Okay, from here to here. This is actually called as the one turn of the DNA. What we call? One turn of the DNA. One turn of DNA. If you count the total number of base pairs present in one turn of the DNA, for aDNA, the total number of base pairs for one turn of DNA is 11. Please write this point. How many base pairs are present in one turn of aDNA? That is 11. Whereas bDNA, if you count it, it should be 10. So 10 base pairs in one turn for bDNA. cDNA has 9 base pairs in one single turn of the DNA. And cDNA is normally not naturally found DNA. It should be synthesized. It is also called as complementary DNA. It is synthesized by taking a natural DNA, separate the 2 strands. Okay, you get only 1 strand. We call it as template strand. And then actually synthetically, that is in vitro, using like lab materials, enzymes, artificial enzymes, everything like artificially. You synthesize another DNA that is called as complementary DNA. You are going to study about it later, don't worry. So it has got 9 base pairs. The zDNA is the largest and a big DNA. Sometimes it is found in the nucleus itself. Okay, so that contains 12 base pairs. In one turn, it contains 12 base pairs. It is largest. Whereas if you take A, B, C DNA, what is common among them is they are all right-handed or right-handedly coiled. So they have coiled, but it is right-handed. So it is right-handed coiled helix. Got it? So this is the thing. Whereas zDNA is left-handedly coiled, left-handed, antiparallel, left-handedly coiled. Whereas ABC right-handedly. This one is Z is this one, left-handedly. Just write the point. Remember, it could be used. Okay, so this is very important. Just remember it. So we are going to discuss about bDNA. So that is actually proposed by Watson and Crick. Done. Okay. For this, very simple. You have already studied about this, but still we recall it. But make sure that you write all the points. These are all very important. In this one, this is you know it. You have been studying the same thing, like double helix model of DNA. You know all the bases. We start. So we know that DNA has 2 strands. What is this called as? This is called as first polynucleotide chain, which is the backbone. That means mainly it contains phosphate, right here, phosphate plus sugar. We know that it's nothing but a polynucleotide chain. Here the phosphate group and sugar acts like the backbone of the DNA. So this is one. We just write. Okay, so this is actually the diagram of the bDNA, double helix, right-handedly coiled. These are all the nitrogenous bases. It has 2 polynucleotide chain, or it has got 2 strands, that is phosphate and sugar as the backbone. That is the first point to you, right? And it has got 5 prime. Normally I pronounce 5 dash, both are correct. 5 prime to 3 prime polarity. One strand is showing 5 prime to 3 prime polarity, one strand. whereas the other strand is showing the opposite, that is 3 prime or 3 dash to 5 dash polarity. Yes, that's it. So these two strands run anti-parallelly. That's it. So two polynucleotide chain or two strands of the DNA are anti-parallel. That's it. So this is the second point. Now these two strands are like normally held together or tightly by the nitrogenous bases or nitrogenous base pairs. We know that for adenine thymine with two hydrogen bonds, make sure that you write this. Whereas for guanine, it is cytosine with 3 hydrogen bonds. So this is the basic. So like that. Normally, if you look into the DNA, so this from here till here, this is actually the one turn of DNA. So remember it, one turn of DNA. For BDNA, how many base pairs should be there? 10 base pairs should be there. 1, 2, 3, 4, 5, 6 are there. Okay, we'll put 7, 8, 9, 10. Now it is a proper BDNA. For cDNA, it should be 9 base pairs. Whereas for zDNA, it is 12 base pairs. For ADNA, it is 11 base pairs, like that. Okay, so this is the thing. Now, if you carefully look, particularly here, this is called as the major group. Okay, so I have got a model. I just forgot to show this to you. Obviously, you know this model. And here, okay, so we'll bend it. Thoda. Okay, so now you could see here. If you look, thoda, the bend here, it's a bit more bended towards the left side, correct? So this is called as the major groove. Whereas here, the opposite of the major groove is minor groove, which is like very it's also bent, but not much compared to the major groove. That's it. So here, it's bit bent towards this side. This side, here is bent. So we say that it is the major groove, and this side is the minor groove. So this is actually double helix model of the DNA. That's all about it. It's very easy. Okay, you have to write around 5 to 6 points. I forgot to tell. The interval, at regular intervals, or we can say that the distance between two nitrogenous bases or nitrogenous base pairs. I'm talking about this base pair and this base pair. It could be adenine, thymine, here guanine, cytosine, whatever it is. The distance is 3.4 Armstrong units. If you count how many base pairs are there, 10 base pairs are there. So, for example, if it is 3.4 Armstrong, like that. So, total one turn, the length is 34 Armstrong units. Remember it, because it contains 10 base pairs and the distance between each base pair is 3.4 Armstrong units. That's it. Done. So, that's all about the double helix model of DNA. Please remember X-ray diffraction, the basis. I forgot. Please add one more point. It is also based on Chargaff's rule. Remember it. That means they proposed this double helix model of DNA based on X-ray diffraction of DNA by Astbury and Rosalind Franklin, and also Chargaff's rule. We have studied it in our previous session. Now we just add one more point. In your notes, please write down for Chargaff rules, whatever the notes you have taken for Chargaff rule, right? Just add one more point, that Chargaff rule cannot be applicable to single stranded DNA as well as single stranded RNA. So it is sure that the Chargaff rule should be applicable to double stranded DNA as well as double stranded RNA. Naturally, you cannot see double stranded RNA, so it is applicable strictly to double stranded DNA. That is this. Hope you understood this. Okay, so chalo.